Right now, all around us, on every single surface, and yeah, even inside our own bodies, there is this bustling, invisible world just teeming with life. We're talking, of course, about microbes. And today, we're going to pull back the curtain and show you exactly how scientists explore this hidden universe, all the way from identifying dangerous pathogens to brewing your favorite wine. Okay, let's get into it.
So what's our roadmap for this journey? Well, it's the source material for this whole explainer, the 11th edition of Microbiology A Laboratory Manual. Honestly, you can think of it as the official playbook for any modern microbial explorer. It is packed with all the techniques and procedures that labs across the entire world are using every single day.
So that begs the question, right? How do you actually go from a simple sample of, say, blood or water or food to really understanding the specific little organisms living inside it? What does it really take to make the invisible, well, visible? And not just visible, but understandable.
Today, we're going to open up a few case files straight from this manual. First, we'll step into the microverse. Then we're going to play microbial detective. After that, we'll hunt for superbugs with some pretty unlikely allies and see how we can put microbes to work for us. And finally, we'll wrap it all up with a look at the complete toolkit for a modern microbiologist.
Okay, our first stop has to be the lab itself. You can think of this place as a portal. It's where scientists take something that is completely invisible to the naked eye and turn it into something they can actually study, identify, and, yeah, even manipulate. Now, here's a really common misconception. You might think you're looking at individual bacteria here, but they're way, way too small for that. What you're actually seeing are called colonies. See, microbiologists put nutrients in a petri dish, and a single bacterium will just multiply and multiply until it becomes a visible mound of millions of identical clones. So every dot, every single streak, that's its own thriving microbial metropolis. One of the most important jobs in all of microbiology is figuring out exactly which microbe you're looking at. And this gets especially serious when that microbe comes from a sick patient. And the whole process, it feels less like biology and a lot more like a detective story.
And every good detective follows a procedure, right? First, they gather the initial clues. They use special stains to see the microbe's shape under a microscope. Then, they profile the suspect, noting how its colonies look and grow. After that, they run a whole battery of biochemical tests to figure out what it eats and what it produces. And finally, they take all that info and check it against Bergey's manual, which is basically the FBI's most wanted list for bacteria, to make a positive ID.
And let's be clear, in a hospital setting, the stakes could not be higher. As the lab manual points out again and again, the goal is speed and efficiency. Identifying the bad guy is the absolutely critical first step to choosing the right medicine and helping that patient get better. This is not some casual academic exercise. It is a race against time.
Okay, let's move on to our next case file. And this one tackles one of the biggest, scariest challenges in modern medicine: bacteria that have evolved to resist our very best antibiotics. We're talking about the infamous superbugs.
This is such a massive problem. I mean, when the drugs we've relied on for decades just stop working, where do we turn? Well, the answer, believe it or not, might not be a new drug at all. It might actually come from a very, very old and very natural enemy of bacteria.
As our source material says so well, the search for new solutions is taking researchers to some pretty unlikely places. And this brings us to one of the most fascinating ideas in the entire lab manual: using viruses to fight bacteria, an idea called phage therapy. I want you to meet the bacteriophage. The name literally means bacteria eater, and that is exactly what they do. These are viruses that have evolved over billions of years for one reason and one reason only: to hunt and kill bacteria. And here's the amazing part. They are hyper-specific. A phage that hunts E. coli, for instance, will completely ignore every other bacteria, including all the good bacteria in our gut. They are perfect little precision-guided weapons.
And this image just shows it so perfectly. This is what it looks like when they win. A microbiologist spreads what they call a lawn of bacteria on a plate. They add the phages and they wait. Every single clear spot you see, it's called a plaque, is a tiny microscopic war zone where the phage army has found, infected, and completely wiped out its bacterial target. It's incredible.
But microbiology isn't always about fighting microbial villains. A huge part of it is about harnessing the power of microbial heroes.
So let's shift gears completely. We're leaving the clinic and heading to the vineyard to see how some microbes act as master brewers, creating one of the oldest and most beloved drinks in the world.
The magic word here is fermentation. Now, it sounds scientific, but the idea is actually pretty simple. You give microorganisms, like yeast, a whole bunch of sugar, and in return, they convert those sugars into alcohol and carbon dioxide. You know, bubbles. It's an ancient process humans have used for thousands of years to make everything from bread to beer to wine.
The lab manual breaks down winemaking beautifully. First, you start with grape juice, which is called must. Then, you add a chemical to knock out any of the wild yeasts or molds you don't want in there. Next, you introduce your star player, a very specific strain of wine-producing yeast. You let it work its magic, first with some oxygen, then without any oxygen at all. And after that, it's just a matter of aging, clarifying, and bottling.
So really, when you see a culture like this, you shouldn't just see a streak on a plate. In the world of fermentation, what you're really looking at is a workforce. Billions upon billions of tiny, efficient, single-celled workers just ready to transform simple juice into a complex, delicious wine.
So we've seen how microbiologists can play detective with pathogens, hunt them with viruses, and even put them to work for us. Which brings us to our final section, a quick look at all the essential skills you need to operate in this unseen world.
The modern microbiologist's toolkit is just incredibly diverse. You've got the basics, of course, like staining and using a microscope. You absolutely have to master aseptic techniques, which is just a fancy way of saying you have to keep everything unbelievably clean to avoid contamination. And then it's got the detective work, the virus hunting, the industrial power of fermentation, and yeah, even genetic manipulation. It's the whole shebang. And this right here, this really brings it all home. On one side, you have the problem, a culture of some unknown bacteria. Could be harmless, could be dangerous. On the other side, you have a tool, like bacteriophages, that can offer a very targeted solution. That's what this entire field is about. It's about that journey from the unknown to the known, from a problem to a solution, by learning how to control this invisible world all around us.
Which leaves us with one last big thought. Microbes have been on this planet for billions of years longer than we have. Billions. We have only just begun to scratch the surface of what they're capable of. So, as the tools in our toolkit get more and more powerful, the real question is, what incredible secrets will they help us unlock next?